Hello, I'm SA, your AI essayist. Please note that all content, audio and video in this presentation are AI generated. As AI can sometimes produce inaccuracies, I encourage you to verify any factual information presented. With that in mind, let's begin.
Philip Showalter Hench, a life devoted to understanding and alleviating suffering. The young Hench embarked on his medical journey, a path initially shaped by the prevailing medical landscape of the early 20th century. Rheumatic diseases, particularly rheumatoid arthritis, presented formidable challenges. Diagnosis remained imprecise, often relying on subjective assessments of symptoms. Treatment options were strikingly limited, leaving physicians with few effective tools to combat the debilitating inflammation and pain that characterized these conditions. Hench's early clinical experience provided a foundation for his later groundbreaking work. He immersed himself in the detailed examination of patients, meticulously documenting symptom progression, physiological changes, and responses to various therapies. His approach emphasized a deep understanding of individual patient histories, recognizing the complexity and variability within rheumatic diseases. These detailed case studies, a testament to his observational skills, provided the crucial groundwork for his future insights. The medical knowledge of the time, while growing in certain areas, lacked a comprehensive understanding of the underlying mechanisms driving rheumatoid arthritis. The role of hormones and physiological processes was emerging as a field of intense study. Endocrine research, then in its relative infancy, offered tantalizing hints of potential therapeutic interventions. Early pioneers in endocrinology, such as those who studied the adrenal glands and their secretions, laid the groundwork for Hench's later work. Their research highlighted the complex interplay between hormones and bodily functions, opening avenues for exploring the potential of hormonal therapies in treating inflammatory conditions. These researchers, particularly those focused on adrenal cortical extracts, provided Hench with a crucial framework for his investigations. Their studies on the effects of these extracts and various physiological processes, particularly inflammation, directly informed Hench's early hypotheses. Hench's work in the pre-Cortisone era represents a period of careful observation and hypothesis generation. He collaborated extensively with colleagues, sharing data and insights. His early interactions with fellow researchers specializing in rheumatic diseases led to a collective exchange of knowledge and experience. These collaborations proved invaluable, fostering a shared understanding of the challenges inherent in treating these complex conditions. The collaborative environment fostered a spirit of intellectual exchange, allowing Hench to refine his clinical approach and formulate more precise research questions. The influence of Hench's meticulous clinical observations on his collaborators' research directions is evident in their subsequent publications, many of which acknowledged his contributions in shaping their research focus. Several researchers, inspired by Hench's insights, shifted their investigative focus towards exploring the potential of hormonal therapies for inflammatory diseases. Hench's early research demonstrated a remarkable dedication to understanding the nuances of rheumatoid arthritis. He recognized the need for a more precise diagnostic framework, moving beyond simple symptom descriptions to incorporate a more comprehensive assessment of patient characteristics. His commitment to detailed patient records, along with close collaboration with fellow researchers, laid the foundation for a more systematic approach to the study of rheumatic diseases. The limitations of the medical tools available at the time, such as the absence of effective anti-inflammatory drugs, only served to sharpen Hench's focus on finding innovative solutions. The lack of precise diagnostic tools spurred him to develop more refined methods for assessing disease severity and progression. His early work was a testament to the power of meticulous observation and the importance of collaborative research in advancing medical understanding. The intellectual climate of the time was characterized by a growing appreciation for the body's intricate hormonal systems. The emerging understanding of the adrenal cortex and its role in regulating various bodily functions, including inflammation, provided a fertile ground for Hench's investigations. This burgeoning field of endocrinology, with its focus on the intricate interactions between hormones and the body's physiological responses, offered a new lens through which to view the pathogenesis of rheumatic diseases. Early studies on the effects of various hormonal extracts on inflammation provided a crucial context for Hench's work, prompting him to explore the potential of these extracts as therapeutic agents. These studies provided a crucial theoretical framework, guiding his clinical observations and shaping his hypotheses about the potential role of hormonal factors in rheumatoid arthritis. His early work highlights the importance of interdisciplinary collaboration in advancing medical knowledge, combining clinical observation with a rapidly expanding field of endocrinology. The influence of these early endocrinological studies on Hench's research is undeniable, shaping his approach and influencing the direction of the subsequent investigations. His early collaborators, inspired by his rigorous clinical observations, further pursued the potential of hormonal therapies, extending the reach of his initial insights. The collaborative spirit and commitment to rigorous observation established the foundation for the remarkable breakthroughs that followed. The stage was set for a pivotal moment in medical history, a moment that would transform the treatment of inflammatory diseases. Philip Showalter Hench's relentless pursuit of effective rheumatoid arthritis treatments led him to explore the burgeoning field of endocrinology. His early research, characterized by meticulous clinical observation and a keen understanding of the interplay between various bodily systems, provided a fertile ground for his later collaborations. This meticulous approach established a foundation upon which groundbreaking discoveries would be built. The limitations of existing treatments for rheumatoid arthritis, a debilitating disease characterized by chronic inflammation and joint pain, fueled his determination to find a more effective solution. His focus shifted towards the endocrine system, recognizing the potential influence of hormones in the complex inflammatory processes underlying the disease. This focus, informed by the endocrinological literature of the time, proved pivotal in shaping his research trajectory. The collaborative spirit that defined Hench's work became particularly evident in his partnership with Edward Calvin Kendall, a renowned biochemist specializing in the chemistry of steroid hormones. Kendall's expertise in isolating and characterizing adrenal cortical extracts provided an invaluable resource for Hench's clinical investigations. Kendall's years of painstaking work isolating and purifying compounds from the adrenal cortex, culminating in the identification of several crucial steroid hormones, directly informed Hench's therapeutic approach. This collaboration was not merely a matter of exchanging information. It represented a synergistic integration of clinical observation and chemical analysis analysis. Hench provided the clinical insights gleaned from observing patients with rheumatoid arthritis, while Kendall provided the means to investigate the potential therapeutic effects of specific adrenal cortical extracts. Their combined efforts accelerated the process of discovery, bridging the gap between clinical observation and chemical analysis. Their work relied heavily on bioassays, a technique employed to measure the biological activity of compounds. These assays, painstakingly developed and refined by Kendall's team, allowed for the precise quantification of the effects of various adrenal extracts and inflammation in animal models. The results of these bioassays provided crucial data for guiding the chemical extraction and purification efforts. The development and refinement of these bioassays, a testament to the technical prowess of Kendall's laboratory, were crucial to the identification and isolation of the active compound. These assays provided a quantitative measure of biological activity, guiding the selection and refinement of the active compounds from the complex mixture of adrenal extracts. This process, a rigorous interplay of biological testing and chemical purification, was instrumental in isolating the compound responsible for the observed anti-inflammatory effects. Simultaneously, Tadeus Reichstein, a Swiss chemist, made significant contributions to the understanding and extraction of adrenal hormones. Reichstein's pioneering work on the chemical structure and isolation of various adrenal corticosteroids provided a deeper understanding of the chemical complexity of the extracts. His detailed chemical characterization of these compounds, achieved through meticulous chemical separation and identification techniques, played a crucial role in facilitating the identification of the active component responsible for the observed anti-inflammatory effects. Reichstein's contributions were a crucial element in the puzzle, providing the chemical framework within which Hench and Kendall's biological observations could be interpreted and understood. His work on the chemical structure and isolation of adrenal corticosteroids was crucial, allowing for the identification of the specific compound responsible for the observed anti-inflammatory effects. The collaborative efforts of Hench, Kendall, and Reichstein culminated in the isolation and synthesis of cortisone, a significant milestone in the treatment of rheumatoid arthritis. Their combined expertise, Hench's clinical acumen, Kendall's biochemical skills, and Reichstein's chemical mastery created a powerful synergy. The successful isolation and subsequent synthesis of cortisone represented a triumph of collaborative scientific endeavor, highlighting the power of interdisciplinary collaboration in tackling complex scientific challenges. This achievement was not a singular breakthrough, but the culmination of years of dedicated research, meticulous experimentation, and seamless collaboration. The detailed chemical characterization of cortisone, achieved through advanced chemical techniques, allowed for its eventual synthesis, making it available for widespread therapeutic use. The synthesis of cortisone was a critical step, allowing for the large-scale production of the drug, transforming it from a laboratory curiosity into a widely available treatment. The impact of their discovery extended far beyond the immediate treatment of rheumatoid arthritis. Cortisone and its derivatives revolutionized the treatment of a plethora of inflammatory conditions. The insights gleaned from the collaborative efforts of these three scientists laid the groundwork for the development of numerous other corticosteroids, each with its unique therapeutic applications. Their work fundamentally altered the landscape of inflammatory disease treatment, establishing a new paradigm for understanding and managing these conditions. The enduring impact of cortisone on medical practice is a testament to the power of collaborative scientific inquiry and the transformative potential of basic research. The development of subsequent corticosteroids, building upon the foundation laid by Hench, Kendall, and Reichstein, further solidified their contribution to medicine. Their legacy continues to shape the treatment of inflammatory diseases, demonstrating the lasting impact of their collaborative efforts. The discovery of cortisone marked a turning point in the understanding and treatment of inflammatory diseases, a turning point that continues to resonate in modern medical practice. The subsequent development and refinement of corticosteroids, all stemming from this initial breakthrough, stand as a powerful testament to the enduring impact of their collaborative work. The discovery of cortisone's remarkable therapeutic potential marked a pivotal moment, yet translating this laboratory finding into a reliable clinical treatment demanded a rigorous approach. Hench's commitment to meticulously designed clinical trials stands as a testament to his scientific rigor. He understood that demonstrating cortisone's efficacy required more than anecdotal evidence. It necessitated a systematic evaluation employing the best available methodologies. This involved carefully selecting patients with rheumatoid arthritis, a condition characterized by debilitating inflammation, pain, and joint destruction. He established clear criteria for patient inclusion, ensuring a homogeneous group to minimize variability in responses. This meticulous patient selection formed the bedrock of his clinical trials, a critical step in generating reliable and meaningful results. Hench's trials were not simply observational studies. They were meticulously planned experiments. He employed a placebo-controlled design, a cornerstone of modern clinical research. This involved randomly assigning patients to either receive cortisone or placebo, a substance lacking the active ingredient. This design, crucial in discerning the true effect of the drug, allowed for a direct comparison between the treated and control groups. The use of blinding, where neither the patients nor the assessors knew the treatment assignment, further minimized bias, ensuring objective assessment of outcomes. These measures, now standard practice in clinical trials, were pioneering steps in Hench's time, reflecting a sophisticated understanding of experimental design and the need to minimize confounding variables. The assessment of treatment response was equally rigorous. Hench developed standardized measures to quantify disease activity, moving beyond subjective descriptions of symptoms. These measures included detailed assessments of joint swelling, pain levels, and functional capacity. These standardized assessments were critical in providing objective data, facilitating comparisons between treatment groups and enabling a quantitative analysis of cortisone's impact. The use of numerical scales, rather than vague qualitative descriptions, allowed for more precise evaluation of treatment efficacy, a critical advancement in the field of clinical rheumatology. The results of Hench's clinical trials were nothing short of transformative. His publications meticulously documented the data, presenting clear evidence of cortisone's remarkable ability to alleviate symptoms and improve the quality of life for patients with rheumatoid arthritis. The data showed a statistically significant difference between the cortisone-treated group and the placebo group, demonstrating the drug's efficacy beyond mere chance. These findings, published in leading medical journals, provided compelling evidence supporting the therapeutic use of cortisone, marking a paradigm shift in the treatment of inflammatory diseases. The rigorous methodology employed in these trials served as a model for future clinical research, influencing the design and conduct of subsequent trials in various medical fields. The statistical methodologies of Hench's era played a significant role in shaping his approach to clinical trials. The development of robust statistical tests, such as the t-test and analysis of variance, provided the tools necessary to analyze the data and draw meaningful conclusions. These statistical techniques allowed Hench to quantify the difference between treatment groups, assess the significance of his findings, and minimize the risk of drawing erroneous conclusions. The application of these statistical methods was crucial in establishing the validity of cortisone's therapeutic effects, demonstrating the profound synergy between clinical research and statistical analysis. This collaboration between medical practice and statistical rigor was a hallmark of Hench's work, setting a high standard for subsequent clinical research. The impact of Hench's clinical trials extended far beyond the immediate application of cortisone. The results profoundly influenced the adoption of cortisone in medical practice, leading to a widespread shift in the treatment of rheumatoid, arthritis, and other inflammatory conditions. His work served as a powerful example of the importance of rigorous clinical trials in establishing the efficacy and safety of new therapies. The meticulous design, standardized assessments, and statistical analysis employed in his trials provided a blueprint for future clinical research, influencing the development of guidelines and regulations governing clinical trials. This enduring legacy reflects the profound impact of Hench's commitment to rigorous scientific methodology. The influence of Hench's clinical trial methodology can be seen in the subsequent evolution of clinical trials in rheumatology and other medical fields. Subsequent researchers built upon the foundation he laid, refining methodologies and developing more sophisticated statistical techniques. The emphasis on randomized controlled trials, blinded assessments, and standardized outcome measures, all hallmarks of Hench's approach, became central to the design of contemporary clinical trials. His work established a gold standard for clinical research, a standard that continues to guide the evaluation of new treatments and therapies, demonstrating the enduring impact of his rigorous and meticulously designed clinical trials. The legacy of his work continues to shape the way we approach the evaluation of new medical interventions, a testament to his enduring contribution to clinical science. The culmination of Hench's decades-long dedication to unraveling the mysteries of rheumatoid arthritis arrived with the 1950 Nobel Prize in Physiology or Medicine. Shared with Edward Calvin Kendall and Tadeus Reichstein, the award recognized the collective contributions to the discovery of cortisone and its transformative effects on inflammatory diseases. The Nobel Committee's citation highlighted the trio's groundbreaking work, emphasizing the profound impact of cortisone on alleviating the debilitating symptoms of rheumatoid arthritis. This accolade underscored the rigorous methodology employed by Hench, a hallmark of his research that extended beyond the mere identification of a therapeutic agent. The committee's decision reflected a recognition of the systematic nature of Hench's clinical trials, his meticulous observation of patient responses, and his dedication to rigorous data analysis. The precise intellectual problem Hench addressed was the lack of effective treatments for rheumatoid arthritis, a disease that caused immense suffering and disability. His unique solution involved a systematic approach to clinical trials, meticulously documenting patient responses to various treatments, including cortisone. A tangible example of this work is his detailed publication of clinical trial results, demonstrating the remarkable efficacy of cortisone in reducing inflammation and improving joint function in patients with rheumatoid arthritis. This publication served as a pivotal moment, shifting the paradigm of arthritis treatment from largely palliative care to a targeted therapeutic approach. The modern relevance of Hench's work remains considerable. Cortisone and its derivatives continue to be essential components of arthritis management, albeit often utilized with a greater understanding of their long-term effects and potential side effects. Hench's work directly built upon the foundational research of Kendall, who isolated and identified cortisone, and Reichstein, who synthesized various corticosteroids. The collaborative nature of their research exemplifies the power of interdisciplinary approaches in scientific discovery. Kendall's chemical expertise provided the crucial compound, while Reichstein's synthetic chemistry enabled the production of sufficient quantities for clinical trials. Hench, in turn, provided the crucial clinical validation, demonstrating the therapeutic efficacy of cortisone in a controlled setting. The influence of Hench's Nobel Prize on subsequent corticosteroid research is profound. The award amplified the significance of the discovery, attracting substantial funding and stimulating further research into the mechanisms of action, potential side effects, and newer, safer corticosteroid analogs. Scientists worldwide were inspired to investigate the therapeutic potential of corticosteroids in a wide array of inflammatory and autoimmune diseases, leading to significant advancements in the treatment of conditions such as asthma, lupus, and inflammatory bowel disease. The Nobel Prize had a significant impact on the careers of many scientists in the field. The prestige associated with the award attracted considerable attention to Hench's work in the field of rheumatology in general. Younger scientists were inspired to pursue research in this area, leading to a surge in research funding and the establishment of new research centers dedicated to the study of inflammatory diseases. The prize also facilitated collaborations between scientists from different disciplines, fostering an environment of interdisciplinary research that accelerated the pace of discovery. The resulting advancements in the understanding and treatment of inflammatory diseases are a direct consequence of the increased research activity stimulated by the Nobel Prize. The award's influence extended beyond the scientific realm. The recognition of cortisone's therapeutic value led to a widespread adoption of corticosteroid therapy for inflammatory diseases. This had a profound impact on the lives of countless patients, alleviating suffering and improving quality of life. The success of cortisone treatment also spurred the development of new drug delivery systems and methods for minimizing side effects, showcasing the far-reaching implications of Hench's work. The widespread use of corticosteroids, however, also highlighted the importance of understanding the long-term implications of such powerful drugs. Subsequent research focused on developing more targeted and less toxic therapies, a direct response to the initial success of cortisone therapy and the ensuing recognition of its potential limitations. This ongoing research reflects the continuing evolution of medical practice, built upon the foundation laid by Hench's groundbreaking work. The Nobel Prize, in its own right, became a significant factor in shaping the course of medical research. The award served as a powerful incentive for scientists to pursue ambitious and impactful research projects. The prestige associated with the prize attracted substantial funding, enabling researchers to undertake large-scale studies and develop sophisticated methodologies. The committee's decision to recognize Hench, Kendall, and Reichstein's work highlighted the importance of interdisciplinary collaboration and the integration of basic science discoveries into effective clinical treatments. This emphasis on translational research remains a cornerstone of modern biomedical research, showcasing the enduring influence of the Nobel Prize in shaping the direction and focus of scientific inquiry. The award's impact continues to resonate within the scientific community, serving as a powerful symbol of achievement and a catalyst for future discoveries. The legacy of Hench's Nobel Prize extends far beyond the immediate impact of cortisone, shaping the landscape of medical research and practice for generations to come. Its influence continues to be felt in the ongoing quest for innovative treatments for inflammatory and autoimmune diseases. Philip Showalter Hench's enduring legacy extends far beyond the immediate impact of cortisone's discovery. His meticulous observational skills, a hallmark of his clinical practice, formed the bedrock of his groundbreaking research. He possessed an exceptional ability to discern subtle patterns within complex clinical presentations, a talent that allowed him to recognize the connection between rheumatoid, arthritis, and adrenal cortical extracts long before the underlying mechanisms were fully understood. This keen observational acumen, honed over years of dedicated clinical work, proved invaluable in guiding his research and shaping his experimental design. The success of his collaborative efforts with Kendall and Tadeus Reichstein further underscores the critical role of careful observation in advancing medical science. Their combined expertise, Hench's clinical insights, Kendall's chemical prowess, and Reichstein's expertise in steroid chemistry represented a powerful synergy, demonstrating the transformative potential of interdisciplinary collaboration in tackling complex medical challenges. Hench's rigorous approach to clinical trials, a hallmark of his scientific methodology, stands as a testament to his commitment to scientific rigor. He meticulously documented his observations, meticulously controlled his experimental variables, and carefully analyzed his data, establishing the gold standard for clinical research that continues to inform the design of modern clinical trials. His work on cortisone, for example, involved a series of carefully designed clinical trials involving patients with rheumatoid arthritis. These trials were not merely observational studies, but rather controlled experiments that allow Hench to demonstrate the efficacy of cortisone in reducing inflammation and alleviating symptoms. This rigorous approach, coupled with his meticulous documentation, allowed him to present compelling evidence supporting the therapeutic potential of cortisone, paving the way for its widespread adoption in the treatment of inflammatory diseases. His meticulous approach served as a model for subsequent clinical researchers, shaping the standards for evidence-based medicine. The transformative impact of Hench's work on the treatment of inflammatory diseases is undeniable. Cortisone, initially derived from adrenal cortical extracts, revolutionized the treatment of rheumatoid, arthritis, and other inflammatory conditions, offering patients significant relief from debilitating symptoms. The introduction of cortisone marked a turning point in the management of these diseases, shifting the therapeutic paradigm from palliative care to targeted intervention. Before cortisone, treatment options for rheumatoid arthritis were limited and largely ineffective, leaving patients to endure chronic pain and disability. Hench's discovery offered a new hope, providing a powerful tool to combat inflammation and improve the quality of life for countless individuals suffering from these debilitating conditions. The immediate impact was profound, dramatically altering the treatment landscape and offering a new level of hope for patients and clinicians alike. However, Hench's contributions extend beyond the immediate impact of cortisone's clinical applications. His work highlighted the crucial role of the endocrine system in regulating inflammation, opening up new avenues of research and paving the way for the development of other anti-inflammatory drugs. His research spurred further investigations into the mechanisms of inflammation, leading to a deeper understanding of the complex interplay between the immune system, the endocrine system, and the inflammatory response. This expanded understanding, born from his initial observations and meticulously conducted trials, continues to shape current research into autoimmune and inflammatory diseases. Further developments in understanding inflammatory processes, such as the role of cytokines and other inflammatory mediators, owe a debt to the foundational work established by Hench's initial discovery. The enduring relevance of Hench's work in modern medicine is evident in the ongoing research into inflammatory and autoimmune diseases. While cortisone and its derivatives remain important therapeutic agents, the understanding of inflammation has expanded considerably, leading to the development of newer, more targeted therapies. However, the principles established by Hench's meticulous observation, rigorous clinical trials, and interdisciplinary collaboration remain as relevant today as they were during his time. The modern approach to drug development, with its emphasis on evidence-based medicine and rigorous clinical testing, is a direct descendant of the high standards set by Hench's work. His legacy is visible in the ongoing efforts to develop safer and more effective treatments for inflammatory diseases, ensuring his contributions continue to shape the future of medicine. The broader implications of Hench's scientific achievements are far-reaching. His work underscored the importance of translational research, the process of translating basic scientific discoveries into effective clinical treatments. His collaboration with Kendall and Reichstein exemplified the power of interdisciplinary collaboration, demonstrating how the combined expertise of scientists from different fields can lead to groundbreaking discoveries. This emphasis on translational research and collaboration continues to be a driving force in modern biomedical research, highlighting the enduring impact of Hench's legacy legacy on the organization and direction of scientific inquiry. His success serves as a powerful example of the transformative potential of collaborative research and the importance of bridging the gap between basic science and clinical practice. In conclusion, Philip Showalter Hench's legacy is one of meticulous observation, rigorous methodology, and transformative impact. His contributions to the understanding and treatment of inflammatory diseases are immeasurable, shaping the course of medical research and practice for generations to come. His work serves as a testament to the power of careful observation, collaborative research, and the relentless pursuit of scientific excellence. The principles he embodied, rigorous experimentation, interdisciplinary collaboration, and a deep commitment to improving patient care remain the cornerstones of modern medical research, ensuring his enduring influence on the future of medicine. His impact extends far beyond the specific treatment of rheumatoid arthritis. It represents a paradigm shift in medical methodology and a powerful example of the transformative power of scientific collaboration. The ongoing quest for innovative treatments for inflammatory and autoimmune diseases stands as a lasting tribute to his profound and enduring achievements.
We really appreciate you tuning into this AI-powered presentation. Our goal is to push the boundaries of AI in content creation, and your input is invaluable. If there's a particular subject you'd like to see us tackle next, or if you have suggestions on how we could fine-tune our AI's creative prompts, please don't hesitate to leave your ideas in the comments section. We're excited to bring you more unique content very soon.